A big pleasure to introduce Dr. Leslie Gordon. This is the first meeting we have of the Tau Biomed Aging, which I hope will become regular and in person. And after the corona, we'll be able to even bring guests from abroad and have half a day with something to eat that we didn't bring from home. But it's a real pleasure to have her start this at the first talk. Professor Gordon did her MD and PhD at Brown University. She's a professor of pediatrics at Brown and at the Department of Anesthesia at Boston Children's Hospital and Harvard Medical School. She's the founder of the Progeria Research Foundation and won many awards, which I won't list because they don't really capture her contribution to the field. She's involved in so many amazing studies that you'll hear, and she's the one person that everyone in the field kind of knows and appreciates and trusts. So even if two labs don't play nicely, everyone knows they can share their most secret results with Leslie, and she always has kind of an overview of what's going on in the field. And she also helps everyone stay really motivated and focused on finding a cure. So without further ado, Professor Gordon.
Well, thank you very much, Danny. That was very nice of you to say. It means a lot to me. It was just so wonderful to get to know you while you were working in the Collins lab, and I'm so glad you asked me to come and speak today. In Israel, and actually Israel is in the news and quite famous, and I actually Google it all the time now because you have the most sophisticated system and highest rate of people vaccinated with for COVID-19, and everybody is in the world is looking to you to understand how they can do a great job and do better and to learn more. So it's kind of exciting for me to be presenting here, but for many, many reasons. And thank you everybody for inviting me. It's a real honor. I'm going to share my screen. Okay. Okay, and is this view okay of my talk?
It's perfect.
Okay, terrific. Well, again, thank you for inviting me. I was asked by Danny to come and sort of tell my story. I'm going to try to do that in 40 minutes or so because I would love to have as much chatting and conversation as possible. And feel free throughout. These are my disclosures. I'm the volunteer medical director for the Progeria Research Foundation, and these are my sources of funding, and I get medication donated to our clinical treatment trials that I'll be telling you about. You know, I don't know that I need to say this to all of you because you really get it, but I'd like to start by just asking a question. Some people may ask, should I really spend time and energy learning about rare diseases? And for many reasons, I say absolutely yes. A rare disease has a prevalence of under 200,000 affected individuals in the US, and that sort of broadly goes out to other countries as well. Progeria is an ultra rare disease, and I'll tell you a little bit about that, but it's extremely rare, so why should we study it? Well, first of all, well, actually I'll save the kids for last, but there are well over 7,000 distinct rare diseases. And altogether, they comprise about 8% of the population. And, you know, I come from pediatrics. Most of the rare children, people affected by rare diseases, most of the time it's genetic. It's going to be there your whole life. And 30% of children with a rare disease will die before the age of 5. So these are very serious things we're discovering every day with all our technologies that we're advancing and the ability to do great genetic diagnostics and exploration. We're discovering that, yes, we're all different from each other, but many, many things are caused by our rare diseases. And it's very important that we understand each of them and how they connect to each other, and also how we can explore and treat them. And there are themes. So today what I'd like to talk to you about is progeria, because to me, the most important thing about studying any disease isn't the number of people that have it, it's just that one person has it. I was taught in medical school that, you know, when you walk into a room with a patient, that patient is the only person in the entire world. And that is true. Each individual child and person matters just so much. And the children with progeria deserve everything that we can give them. And that's what Danny has helped to do, and I'm here for, and all the people in the field of progeria. But if for each of you, each field that you're studying is very important too, and you might just be feel like you're at the bench and not connected to whomever you're trying to help, but that's not true at all. We all stand on the shoulders of basic scientists. There would be no clinical trials or treatments without basic science. So today what I'd like to do is tell you about progeria, about the disease, phenotype, what it looks like, talk about the biologic and genetic basis of disease, and key aspects briefly of how this may overlap with aging and why that's important. And then I'll describe the things that have led to the clinical treatment trials and the results of those trials and where we're going from here. This is Rachel. This is Rachel when she was born and as she grew up. And here she's about 9 years old, 9 or 10 years old, and she actually passed away shortly after that. But she's gorgeous, she's beautiful, and she had progeria. Progeria is a segmental premature aging disease, meaning it has some overlaps with aging and some things that does not have in common with aging. And each field can inform each other. It is generally not passed down. It's an autosomal dominant sporadic mutation that occurs. We know that now. We didn't know that when we started the foundation. The children without the recently approved treatment have a lifespan of about 14.5 years with a pretty broad range, but all die very young. And they die of cardiovascular disease, atherosclerosis leading to heart attacks, sometimes stroke. This is a multi-system disease. There are a lot of systems affected, and we look for what unifies this disease biologically in our investigations. Some systems are not affected. The myocardium, the neurologic system, the kidney, the liver, immune system. So this, in fact, for our clinical trials is very, very lucky. But we look for the similarities and differences between the systems that are affected and are not affected to try to understand the natural history. And these are snapshots of what we see clinically. The children fail to thrive in a pretty dramatic way, although they're born looking normal. They gradually lose hair, all of it in their first year or two of life. There is a tremendous global lipodystrophy. And so they look very muscular, although the muscles are really small. But that's a big challenge for us in many ways. There are joint contractures, as you can see here in the hands and the knees, nail dystrophy, skin signs, bony signs. There's a lot of, on x-ray, you can see a lot of bone effects of progeria. And of course, the cardiovascular disease that we've been talking about. So here in the white, this is an MRI showing lots of stroke. And then here, these are carotid arteries, where in a 5-year-old, on this side, you see this is the artery in the neck coming up, lots of blood, nice flow into the brain. And on your left side here, you see a complete blockage for the other side of the brain. And this is part of what we are trying to combat. Well, in 1998, our son Sam was diagnosed with progeria. He was almost 2, and my husband and I, being clinicians, and I was a clinician scientist, we immediately, but just as parents, dropped what we were doing and tried to find out what was going on. Did we have any place to go? Who was working on this? Was there a treatment? And this is what happens to parents of children with rare diseases all the time. they drop everything, they try to find out what's going on with their child, and often the story is similar to ours. These days, maybe not as often, but we still hear about it all the time. There was nobody working on progeria. 2 or 3 scientists had ever worked on progeria. There was no research funding. We didn't really even know if it was a genetic disease. And essentially, there was just nowhere to go, no treatment prospects. The physicians didn't know anything about it. And so we did what we could. We did what parents of children with rare diseases do many, many times, scientists, physician or not. We founded the Progeria Research Foundation, whose mission was and is to find the cause, treatment, and cure for progeria. And this is Sam, when he was a little bit older, and my husband Scott, who had not only an MD, but also a Master's of Public Health. So he helped with the structure of the organization. And so together with my sister, who was a lawyer, and our colleagues and friends, we formed this organization. And this is how we started things. We just said, what do people need? What is it that's stopping you from doing progeria research? And where are all the patients? So the first thing we did, and what I'm showing you here is going back, this is a flow chart of what we could do at the beginning, not knowing if this was a genetic disease, not knowing much about the biology. We started the things that we thought might impact the field. Of course, everything starts with the patients. So and today still, we search for patients and children and families, because you can't help them if you can't find them. So we initiated an international progeria registry, which today is invaluable, and you'll see later on was invaluable to the currently first ever approved drug for progeria. So that's very important for communication. And we also, for scientists, started a cell and tissue bank, because scientists said, we don't have the cells and tissues that we need, and we don't have the clinical information. So we started a medical and research database. And these were the first sort of feed-ins, and they live today. They're important today to the field. Scientists from around the world use them. And then, a few years later, we reached out, and we actually had our very first workshop. And from there, we met Francis Collins. We met Francis. Francis met Sam. He had long been interested in a patient called Meg that he had in his clinical practice as a geneticist, that he was just so frustrated he couldn't help. And then Sam came along. They became fast friends, and he got involved. And then we formed something called a genetics consortium, like 20 scientists looking for the gene mutation for progeria. And Francis Collins' lab led the way. Just 10 months after we formed that in 2002, his lab, Maria Erickson was his postdoc at the time. We discovered the gene mutation for progeria. And really what's said on the bottom of this page is true. We were catapulted into a new phase. This allowed us to do so much that I will show you now. So what did we discover? And you probably all know, we discovered that Lamin A is the culprit, an internuclear membrane protein that has both cell structure and broad cell function effects. Not a lot was known about it, but something was known about it. Lamin A is expressed in differentiated cell types. And that makes sense, because children with progeria are born. They're born looking fairly normal, which means that the early cells, the immature cells, don't express it. And as the fetus grows, it starts to express the culprit, the abnormal Lamin A, which I'll call Progerin. And this is the mutation. A single base change. One, very consistent. 90% of children with Hutchinson-Gilford progeria syndrome, progeria, have this base change. A single base change in exon 11 of Lamin A. Here you see a consensus sequence for a splice site. This is what you and I have. This is our splice site. And here is what children with progeria have. A C cytosine to a thymine leads to a silent mutation, so the same amino acid code, but opens up or makes more available a splice site. And so what you're seeing here is just a crude cartoon of exon 11 and exon 12 of Lamin A. Here's a normal splice, but this is what happens in progeria. 150 bases are chopped out where they're not supposed to be. You get a shortened protein of 50 amino acids shorter. And what you see here is just a Western child, mom, dad. This is Lamin A, this is Lamin C, and here is Progerin band only in the child. And so what could we do? Everything in green is what I showed you before. Everything is in red is what we could do once we understood what the gene mutation for progeria was and had discovered that what we call progerin, that abnormal disease-causing protein. We could then run diagnostic testing, and we do that today as well. It's done throughout the world, but many children don't have access to it. So in many countries, it's so rare that there isn't a diagnostic test, and physicians send the blood, and we have a diagnostic testing program. The Collins lab and other labs produced a mouse model of progeria, which couldn't be done before. And I'm sure you can see how incredible that is, because we can, because we can test drugs and things like that. The link to aging became much more accessible because we could test for the presence of progerin and ask questions in aging individuals about progerin and this abnormal Lamin A. And so in asking the questions about aging, which by the way, I had never banked on aging. I said, well, maybe it looks physically like aging in some aspects, but biologically we have no proof. And why draw from a field that may or may not be able to help children with progeria if we really don't know there's a link to aging? But we looked for progerin, the abnormal Lamin A, in aging individuals, and a couple of really nice studies were done. This is from Karima Jabali's lab. This is a skin biopsy, and she created a monoclonal, an antibody that's specific to progerin, doesn't overlap with Lamin A, and each one of these red dots is a single cell nucleus staining for progerin. And it increases as you age. So if I were to show you a 16-year-old, a 15-year-old, a 14-year-old without progeria, this box would be black. So this is a forehead of a 93-year-old. So this was quite amazing to us because we had no idea how frequent the presence of progerin would be as we age. This is in the skin, this is in the vasculature. This is Michelle Olive and I and Betsy Nabel did this study where we looked in the vasculature, the adventitia, the media. Here is plaque. On the x-axis, you see age, and you see the levels of progerin going up about 3% a year as we age. Of course, it's 100-fold less than those with progeria because those with progeria have this fatal disease. So finally, a biological link to aging, which was really, I think, very exciting because then we knew that the aging could draw from progeria, progeria could draw from aging, and I think that really just helps everybody. And of course, begs the question, if we can get at progerin, can we also get at some aspects of generalized aging? Okay, I told you a little about the diagnostics program. That's one of the ways that we find not only recruit but involve children in our clinical trials today. So it's really, really important at sort of going through the registry and into that program. We are still funding preclinical research throughout the world, and these mouse models exist. The Collins mouse model, the Collins lab mouse model, people have all over the world in their laboratories. It's a back transgenic model where the human progerin is in the mouse. The human mutation for progerin is in the mouse, and it's progerin producing, which is really important. There's another one where Carles Lopez Otin of Spain made a beautiful mouse model, and that's that's a little bit made a little bit differently. The mouse itself has the mutation, and that produces progerin as well. And so we've sort of spread all of this out and have now know of about 135 children with progeria. Now the population is 1 in 20 million, so we know there are about 400 children in the world, and we're always in search of those children, but we know of 135 today. And that's the pool of children, and that we target for things like our clinical trials, and we keep in touch with all the time and keep them informed about what's going on in the research field. Now, what we did once we understood more about the genetic mutation and what the problem was with LMNA, we were able to pull in a small but very, very important group of LMNA scientists to talk to us about things like post-translational processing. Now, on the left, what you see here is normal pre-LMNA processing. There are 4 steps. So LMNA is peeled off of the RNA as a pre-lamin, and then it is farnesylated in this first step, which is very, very important because a farnesyl group is placed on to the 5' end, and that is a lipid-loving side group, such that the molecule can intercalate or associate with membranes. And many proteins in the body, about 200, are actually farnesylated, and they use that to associate with membranes, like the internuclear membrane here for progerin. And then after that, the AAX comes off. There's a carboxymethylation step, and the entire end is cleaved so that the farnesyl group comes off here for mature normal lamin-A, and then the cell divides, and there's a process where lamin-A is gotten rid of. But progerin is different because it's missing these 50 amino acids. It's able to be farnesylated here. But the attachment site for the ZIMSD24, the enzyme that cleaves, is missing. So it can be carboxymethylated, but it can't be this last step defarnesylated, which also happens with ZIMSD. So what you have is a shortened, sort of abnormal protein that still associates with the internuclear membrane, but is not, but remains permanently farnesylated. And so not only is it toxic, but it stays. It doesn't turn over like the lamin-A does. And that's going to be very important. So notice this process, post-translational enzymes used, farnesyltransferase, and we took advantage of that because there was a drug category produced called farnesyltransferase inhibitors. Because the farnesyl group is used with so many proteins, it's also used with something like RAS cancers. And Schering-Plough and other companies had been developing farnesyltransferase inhibitors for those cancers. And so what we did was to sort of repurpose that and say, well, could these drugs also help for progeria? And here's the evidence that that is true. And this comes from lots of labs, mostly the Collins lab, where I'm showing, but also Steve Young's lab, where I'm showing you a couple of things. If these are different types of farnesyltransferase inhibitors that are being used, but this in a sum is our preclinical evidence that led us to clinical trials. Here you see the nucleus of a fibroblast, normal fibroblast, a fibroblast with progeria. They're characteristically blebbed and evaginated. But treating with a farnesyltransferase inhibitor normalizes this. So in vitro, this was our first evidence that this might help progeria. In addition, what you see down here is a mouse model, not the one I told you about before, but a different one in Steve Young's lab, treated with a farnesyltransferase inhibitor, much bigger and healthier. This is the sick mouse, untreated. Survival increased about, I would say, 20% in the laboratory of these mice when they were taking a farnesyltransferase inhibitor. And here you see vasculature. This is the aorta, a slice of the aorta. in the Progeria mouse model. This is wild type, a normal wild type mouse aorta wall stained. You see what you see, you're seeing the blue. These are smooth muscle cell nuclei, nice and healthy. In Progeria, the smooth muscle cells, with time, they fade away. They become sick, they die off, and the vasculature becomes very abnormal. There's a lot of extracellular matrix, not a lot of smooth muscle cells in the media. And this is the treated mouse, looking very much more like the normal aorta. And with all of this preclinical evidence together, we then launched into our first set of both natural history and clinical and a clinical treatment trial. We did them simultaneously because it's such a rare disease that that we really couldn't afford the time to do them sequentially. So we started a weighing program from home. Children would weigh themselves, and we wanted to ask, would that trajectory change with treatment along with other things? And finally, we were at the stage in 2007 where we initiated a clinical treatment trial. And we did this at Boston Children's Hospital, a single site open to children all over the world. And we flew in the families, flew in the children for a good reason. We wanted to have all of the children's tests done by the same people longitudinally over time, so we could best ask whether the treatment was working. We didn't know much about objective measures that we could say definitively would change with treatment, or that we even knew the trajectory of, so that we could, measure and ask if a treatment was working. So we took the careful approach. We flew everybody to the same site. In the first trial, every 4 months to be evaluated. And we brought them in pairs because they spent a week in the hospital and it was pretty difficult for them. And if they had each other, they were happier and did better. And it was actually pretty glorious to see everybody every week sort of running down the hall, the kids running down the halls and meeting each other and being happy with each other. It was really, really neat. So that was initiated in 2007. And in 2000, it was a 2-year trial. We filled our recruitment in 4 months because we had done a lot of pre-work, and that's where the registry came in and communication with the families. And what did we find? Well, we found 4 things that we thought were improvements in body systems. The rate of weight gain very modestly improved. It wasn't a lot, but it was a little bit. It was enough to satisfy our prospectively designed primary outcome parameter. But more importantly, the cardiovascular stiffness was improved. And I'll tell you a little bit about that in the next couple of slides. Their bone structure improved. And we evaluated that with a micro CT. The structure itself improved. Now, kids with progeria don't have bone breaks more than any other population, even though their bones are very small, but they do have structural abnormalities. And also their neurologic hearing improved. But importantly, some things didn't change. Their bone mineral density, which is not terrible. Like I said, they don't break bones sporadically, but that wasn't improved overall. We didn't get to the lipodystrophy, the joint contractures, the hair, the dental. So I guess what I'm feeding into here is that we have something that could be a treatment, but certainly we have a long way to go for cure. And we need to sort of try to understand what body systems we can get to with which medications, and what that all means for the disease. What I'm going to show you here is the cardiovascular changes that we found. This was really, really groundbreaking for me, at least. Children with progeria, it turns out, have an atherosclerosis. They also have which has plaques, the traditional plaques that you hear about, and also their blood vessel walls are very, very stiff. Now, this happens to the rest of us as we age, anyways, but in progeria, it happens very, very quickly. And the way that we measure, and what I say to patients is, it's like if you have a stiff pipe, and you have to get blood out to your body. And the stiffer your pipes, the stiffer your vessels, the harder it is for your heart to pump that blood out to your body because it has to pump an adequate amount. And the way we measure vascular stiffness is through something called carotid femoral pulse wave velocity, the speed of travel of blood from the neck area to the thigh area. And that will, and so therefore a high number, a high pulse wave velocity, a high velocity means that your pipes are very stiff, your vasculature is stiff. You want that to come down lower. And importantly, in the aging population, a decrease of 1 meter per second correlates with a lower incidence of fatal heart attacks in the rest of us. So this is a very important measure of your cardiovascular health. And this is what we found in the children with progeria. They had a dramatic change. They started incredibly high. This is the normal range for children way down here. Some of these children went way up near 20 meters per second. I mean, these are all very abnormal, and almost all of them went down by at least 1 meter per second. So that was very enlightening to us. We saw for the first time that we can actually get to the cardiovascular system in progeria. This is a picture of the ultrasound of the carotid arteries, another demonstration of the stiffness of the vasculature. The black is blood. This is the wall of the artery. This is you and me, normal. Children with progeria, very bright, very echo dense, we call it. But with treatment, they backed off and came in much closer to the normal range. And these are the graphs of representing the stiffness, the amount of white that you see here. So these things are sort of all telling a story and marrying to one another. And here I'm just showing you physically what happens in progeria. If you don't have progeria and you get cardiovascular disease, your adventitia, you see, so intima, media with the smooth muscle cells, and adventitia is nice and thin. But with progeria, it's very, very, very thick. And that is also part of the stiffening event. So this was great for us. But when we went to the FDA and we said, look, we think we really have something here. What about this carotid femoral pulse wave velocity in the vasculature? They said to us, it's very difficult to transfer what's known for aging to what's known in progeria. And they really said, you know, I don't think it's enough that we could get approval on this, but they liked something that we had already been doing, which is looking at survival curves in progeria and looking to whether or not maybe treatments could help with survival. This was a little complicated. We had open label clinical trials because all of the children we felt deserved the treatment, and you know, it was an ethical decision. Oftentimes in pediatrics, that's what we do. However, we had this international progeria registry in our medical and research database programs. So we started to take a look, in response to that, we started to take a look at whether or not we might be able to assess survival. Now, you know, it was a 2-year trial. The kids lived 14.5 years. That's a tall order. And so it took us a while to do, but I'm going to show you the results of that. Our first task was to look at children that weren't treated with Lonafarnib over time and concurrently, you know, in the day and age we were working with the children as well. And this is the survival curve that we came up with using several hundred children, several hundred cases. We found, this is where we found our 14.5 years. And as you see this survival curve, survival is here on the y-axis, age of the children. It's pretty normal and regular, and this told us, okay, we have something to compare to, and this hadn't been developed yet. So what we did next was ask, well, what happened to the children in the trials? And what you see here is for each drop is one child passing away. This is the children in blue did not get the treatment. Again, it was an open label trial, but there were some children that we didn't know about at the time of the trial, and we were doing this years later. So we hadn't identified them, but then when we did, for example, we could use this tracker for the untreated group. And so there were 9, over the period of the treatment trial, 2 years, there were 9 deaths in the untreated outside of trial group, and there was 1 in the trial. And so that correlated to an 88% reduced risk of death. But in addition, with some other calculations, the FDA determined that we at least had 2.5. We updated this as well to 2019, and they found an extension of 2.5 years of lifespan. And we'll keep updating that over time. But that's about 20% of the lifespan of a child with progeria. And although it doesn't bring them into late adulthood, a lot of the children are entering adulthood now, 18, 19 years old, and it's really exciting to see them come into their own. But of course, we have a long way to go because we want to find additional treatments and the cure. But this is our start, and it's really exciting, and it led us to FDA approval. So in 2018, armed with this information about survival, and as happenstance, we had been getting the drug for the trials from Schering-Plough, and then Merck bought Schering-Plough, so we were getting it from Merck, but there was a company called Eiger who desired a license to Lonafarnib for a different indication called hepatitis delta with many more people, so it might be much more profitable for them. But we had this late stage data and they partnered with us. And in November of 2020, we got FDA approval. Our very first drug was approved for Progeria. And what I tell people is, this sort of launches us into a new world. Only about 5% of rare diseases have approved drugs. Now, we want that to be much, much higher, but hopefully this kind of a story can help others as well. Very exciting, but we have an awful long way to go. So Lonafarnib is our first treatment. It tells us we can push this treatment towards health. But what this really does for us is to say we can push it. We can push it, and we have to push it more. We have to find new treatments. We have to find a cure. And that's what we've been sort of layering in with this all along, which is essentially that by funding laboratories, providing materials, and collaborating with brilliant, motivated scientists throughout the world, we will really be able to get there with new treatments and the cure. And I just want to show you where we are with a few of those. This is a picture showing you one of the more recent papers, but the next on DNA base editing. But when I sort of look at the field overall, what I'm showing you here is the most promising potential trial treatments for the next, say, 6 months to 2 years. There's a lot going on in the background. There's not even in the background. There's a lot going on a little bit earlier stage things that we fund, that NIH now funds, that others are doing, like Danny, to say, well, are there other treatments, other ways and methods to find new treatments that we can bring into the picture. So there's a lot going on in earlier stage, but here we have the later stage items. And what's interesting here is one is a small molecule, and each one of these graphs shows you what's happened in the mouse models. Now remember, Lonafarnib was able to increase lifespan of the mouse models at most 20%, but today with the models that we have, it doesn't budget much. So in that regard, it worked in the children to enough of an extent to make a difference, and it worked in the mice modestly as well. Here we have a small molecule called Progerin that was produced with funds from PRF to a lab of the Park Lab in South Korea, and he's done just an amazing job bringing this along. And what you see here is it's about a 10-week increase in lifespan for the Collins model. It's called G608G homozygous mouse. And so that's a nice curve, and it's a small molecule, it's oral, and so it's terrific, and hopefully that'll be coming along into trial. But also interestingly, we also have an advanced RNA therapeutic, Morpholino, that we're working with that does an even better job of increasing the lifespan of the mouse models for progeria. And this particular RNA therapeutic is being adapted. It was given IP in the mice, developed for IV infusion, and we're now developing it for subcutaneous injection into the children, because that's a feasible thing to do. It would be about a once a week injection, a small injection that would actually have the benefit of avoiding the gastrointestinal system and be implemented through an insulin syringe. And then finally, we have this, which is the most sort of cutting edge DNA base editing. This came out, so I should say this RNA therapeutic graft came out of the Collins lab. Beautiful, now what's now almost an 8-year investment of their skills and collaboration. And here, David Liu's lab at the Broad, along with the Collins lab and PRF, collaborated on this DNA base editing project, where literally he uses an AAV to edit the C to T change from the T back into the C. That's the DNA base editing project. And the mice live almost into old age, like 2.5 times longer. So this one is really, really cutting edge, really new, but we really think it has great potential over the next couple of years. I just wanted to sort of highlight a couple things about Sam. He was with us until 2014 and was able to create really an amazing movie that you can get on HBO Go called Life According to Sam. And it's not just Sam, it's the other kids too. It chronicles, in essence, him as a human being, which I think he was pretty amazing, but also the clinical trial, that first clinical trial. So it's very exciting to see. And he gave a TEDx talk when he was 17. And that talk has been viewed by over 100 million people. I think it's the second most watched talk in the world on the TED Talk, on the TEDx site. So it's really exciting. But the things I thought were pretty amazing and really hit home for me were what we're fighting for. His philosophy for a happy life was to be okay with what you ultimately can't do, because there's so much that you can do. That coming from a 17-year-old is pretty amazing. But I've seen this kind of wisdom from many, many children throughout the world with progeria. They are just wild, and their participation in these trials is absolutely courageous. Surround yourself with people you want to be around. I think we all want to strive to do that, be around quality people. Keep moving forward, which is exactly what we're doing here all together. People throughout the world like Danny and others just moving forward to help these kids. And most importantly, he said, never miss a party if you can help it. So this is, again, Sammy and Scott and myself, my husband Scott and myself. Thank you, Danny, for inviting me. I can't begin to list the number of collaborators involved in all of these different phases of success for progeria. I work at Boston Children's Hospital as an investigator for the clinical trials. The team is just world-renowned, unbelievable people striving every day since 2007, every day, to understand progeria, to run successful clinical trials. But again, all of the basic scientists that are working on progeria are very, very important to these kids. And thank you again. And I'd love to chat more and take any questions you have.
Leslie, thank you. That was absolutely remarkable. I'll be fielding the questions for you, if I may. And first of all, the personal story is, of course, so inspiring and heartening. We just, on a note on what we're doing, we're working with Dr. Moran Rubenstein here at Tel Aviv University on GRIN2D, and also working very closely with a family. And so we can certainly relate to some of the issues of starting, Keith Duncan started Cure GRIN a couple of years ago, and so much younger and much earlier than where you're at. But again, the importance of studying a rare disease and how many people it really impacts, and bringing the scientists in to be part of the story and the clinicians really makes such an important impact. So thank you, that was really remarkable. Okay, so why don't we open the floor to questions, and you can unmute yourself, you should be able to, and ask a question.
You know, just in response to what you were saying, that is a beautiful way to end this, because people are starting foundations all the time, parents, and they're reaching out to each other and saying, how did you do this? How did you do that? And I have the opportunity, and many people helped me along the way. And now and still are. And I have the opportunity to help others who are starting out as well, and to say to them, there are many different ways to do this. And the ways that we did it are not the only ways, but I think you can do many things more easily than we did, because 20 years ago, to start a cell and tissue bank of your own was unheard of. And today, you just have to pay a company to do it for you. I mean, and the technology, discovering the gene mutation for progeria took so long. I mean, Francis Collins was literally not finished sequencing the human genome when we were working together on this. And today, what we couldn't do in years, you can do in weeks. So I think that there is a lot to be learned from each other, from all of these advances. And you're right, bringing in the scientists is crucial, and the clinicians is crucial as well. So thank you for that comment about others who have started their own organizations. It's really important.
So do we have any questions? You have actually guests from all over Israel who've joined today. So while somebody is gathering a question, I wanted to ask you, what is the access program, the managed access program that you had written on one of your slides?
Oh yes, I'm sorry. So what's really nice that's happened is, so we ran this first clinical trial, and then we ran dovetailed that with a second clinical trial. We added in 2 medications to Lonafarnib, a statin and a bisphosphonate. And then at the end of that, we didn't have another clinical trial to dovetail, but wanted to keep everybody on Lonafarnib. So the FDA and our institutional review board allowed us to keep everybody on just the Lonafarnib as an extension of the trial. And we've done this several times. And so the only way to keep children on Lonafarnib from 2007 until just a few months ago was through this clinical trial. And we just kept going and going and going. What Eiger Biopharmaceuticals did once they came in is started what's called a managed access program. And essentially what the FDA allows you to do now is they've expanded what they used to call their compassionate use program. The drug company has a protocol approved by the FDA, and also it can apply to many other countries, not all, but many, where children could start getting Lonafarnib through their local physicians. So many children with progeria came off our basically perpetual clinical trial and into that program where they could just get their medicine through the local doctor. And then that served, at least in the United States, that served until approval, when they could get some commercial supply outside of the United States. In Israel, in fact, it's not approved, but the managed access program applies. So there are children in the Middle East and in many countries throughout the world getting their medicines off of the clinical trial through that program.
Wonderful, thank you. Okay, we have a bunch of questions. We'll start with Anat Ben-Zvi, who's visiting us from Ben Gurion University in Beersheba. Hi, Anat. You are on mute, even though I believe you can unmute yourself. You still can't? Okay. Let's see here, what's going on? Can anybody else unmute themselves?
Yes.
You can. Okay,
Yeah, we
so
can.
Anat, it's just you. Okay, so why don't we, until you figure it out, Yossi Shilo from Tel Aviv University has a question.
Yes, first, thank you for a really breathtaking seminar. I don't know when was the last time I said that seminar was breathtaking, but this one certainly was. The question is with regard to future treatments, the ones that are now being tried on mice based on oligonucleotides and so on, and other methods. I was just wondering, when it comes to the patients, which tissues will be actually targeted at what age? In other words, the question relates to the very basic question, what are the original or the cells of origins of all this?
Yeah, you just in one moment hit upon one of the central and most difficult issues that we have. So far in our clinical trials, we have chosen drugs that pretty much get into every cell. We don't need it in the brain tissue, because the children have normal intellect and their neurons do well. But we haven't been able to target necessarily the cardiovascular system specifically. I could tell you that what tissues are we most interested in? We're interested in the cardiovascular tissues, the media and adventitia, but even the intima. So if we could get to that, the cardiac tissue itself, the myocardium, it's not severely affected, but I think it could benefit. The children do get some, also their valves get diseased as well. They get plaques and calcium and things like that. But also we haven't been able to affect the fat cells. These kids have insulin resistance. They don't get diabetes, but they have insulin resistance. And I think if we could get their fat cells to be healthy, their lipodystrophy solved, they could actually improve their cardiovascular disease as well. So I don't think it's just the vasculature involved. I think the lipodystrophy solving that would be really important as well. And of course, for quality of life, you've got joint contractures due to this terrible extracellular matrix problem, the collagen, the elastin, it's all disorganized and inappropriate. And so what happens with these medications? Do they affect everything? Can we target them? So this is what we've done. We haven't been able to in a precise way, target. But what we've done is, and what the labs have done, is when they analyze the mouse models for treatment, they look at the cardiovascular system. They say, are we getting to the vasculature? Because if you give a mouse model a treatment and it lives longer, but that treatment doesn't get to the vasculature, will that really help the children? And so we know that for all three of these potential medications, potential trial medications, they are getting to the mouse vasculature. And that's about the best, and living longer, but that's about the best we can do. They get to other organs as well. How much those organs play a role in treating disease, we don't know.
Leslie, can I just ask you to stop sharing so that we can all see each other while we're asking questions? And now Anat can unmute yourself. Sorry, I had to do something. Press a button. Go ahead, Anat.
Thank you. Here I am. Hi. Well, first, thank you. It was exciting. I have 3 kids on the autistic spectrum. It's not a rare disease, but I understand the challenges.
Very important.
Some of them, I guess. I wanted to ask about the DNA editing direction, and what ages can this be applied for the mouse?
Oh, that's right. That's right. I didn't address the age issue. I'll do that now in a couple of different ways. The publication shows that we injected it at 2 times, P3 day 3 retroorbitally, and day 14 IP intraperitoneally. Now, it's at 3 days the mice are really, really small, and it was really, really hard to do. Even though that was a successful graph, and they had some significance, it was really the P14, which is, just very, very young still, if you want to correlate with humans. And that brings up the question of, well, can you reverse disease? But I'll hit that in a moment. What we have done for the clinical trials is we have not restricted anybody of any age from coming in. So yes, in a natural sense, you would think the younger the better. But what we found in our trials is that children, and I guess you'd say young adults, can improve at any age their vascular stiffness and other elements. And so, you know, for a child who's 12 or 13 years old and they have, I'm bouncing off this as you know, because we've talked about it before, but many things, including the carotid femoral pulse wave velocity, it's going to be very high. But they have a long way to drop, and they were able to improve. And objectively speaking, more than somebody who was 4 or 5 years old, because they just didn't have as much disease. So we were able to help children at every age that we were allowed to enroll from 12 months on. And I would foresee doing that again, giving everybody the opportunity, because we just really don't know if the drugs will be able to help somebody at any age. So did I miss part of your question?
Well, I guess one of the reasons it's critical in my mind is that many other diseases that are single base changes could be, if this approach works and you can apply them later in life, then there are many rare diseases though.
I mean, that is exactly right. You are right on target. Why stop yourself? Because children are pretty pliable, right? I mean, you think, I mean, I cannot tell you, when I saw not only it's very hard, not knowing the natural history of disease very well, it's very hard to say something has stalled in its progression, right? Because you need a trajectory curve. But it's a lot easier when something improves. And I was completely, I had none of us cardiologists had any idea that this feature could improve. And so I wouldn't sell the kid short. I think it's our responsibility to just do a really good job with our measuring and testing so that we can see what might be there. But I wouldn't inhibit any you know, by age, but that, you know, certainly presents challenges when it comes to trial design.
Anybody else have any questions?
I want to ask something along this question. When is actually the first diagnosis? What age? What time?
That's a great one too. So, when we first started diagnosing with the diagnostics, I mean, progeria was so poorly known in the world that kids were getting diagnosed really, really late, if at all, you know, even 9, 10 years old. Today, that still happens, you know, because progeria is so rare. But a lot of times we get kids earlier, and it's the dermatologists, because they're such visual people, that often recognize some of the skin signs, which are pretty unique actually. And so if somebody has seen progeria, it's kind of individual. And so it's in, you know, it has a lot of sort of very characteristic features. If they've seen it, then bells go off in their head and they diagnose earlier. So our clinical trials now involve a lot more kids age, you know, 4, 5, 6, and even younger. And diagnostics as well, even though we find also, we do find children at older ages. And I think what's really helped is the number of publications. I mean, we're in every medical school classroom now as genetics has advanced. This is a genetic disease that has been presented. I never learned about progeria in my medical school career. I never knew what it was until Sam was diagnosed. So earlier, yes, but still also some kids later as well, depending on where they come from in the world.
Okay, do we have one last question from anyone? No, okay, so Danny, you want to wrap this up?
So thank you very much for coming and talking to us. I'm sure there'll be a lot of questions, and maybe we'll talk about some of them internally. And if something pops up, I'll be happy to ask a question. Can you hear me?
Sure. Who's asking the question?
Hi, I'm Vijagisha.
Hi. Yes, please go ahead.
And probably it's a very stupid question, but I wanted to know if there's a way that we can diagnose this within the womb itself, by some sort of, tests or anything without, I don't know, like without the baby being born, before the baby is born? Can we somehow get to know by Western blots or anything?
You can, a prenatal diagnosis is possible. So there are some earlier tests and a little bit later tests, but this is something that OBGYNs do all the time for other diseases as well. And so when I counsel parents, there are in any genetic disease that is not passed down, there are still some cases of what's called mosaicism. So if you have a child with progeria already, your chances of having another one, it's no longer 1 in 20 million or 1 in 10 million live births for incidence. It becomes more of like a 1 to 2% chance that you'll have another child, because one of the parents might have what's called mosaicism, and so some of the sperm or egg might actually have the genetic mutation. And so some of the parents have gotten a prenatal diagnosis, and you can do that with your OBGYN. Now, actually, you might even be able to do it with blood tests. I'm not sure, but usually it's a needle that goes in, and this is something that can be done pretty easily, pretty low risk.
Okay, thanks.
Thank you. And certainly as whole exome sequencing becomes moves into the clinic in routine genetic diagnosis, but that of course opens a whole other can of worms in terms of ethics and certainly complications, and very limited in terms of places in the world that would do that type of genetic diagnosis. But we will get there one.
You bring up a whole new discussion. I mean, for somebody coming from where I'm coming from, for $1,000 right now, you can do whole exome sequencing. And I just, I think in progeria would never be on a sort of a traditional, it's so rare, on a traditional screening test. But if you're doing that for every baby, you would catch it, and you would catch it earlier, and you'd catch it for all the other 7,000 rare diseases as well. And I think that could really do some very, very good things as far as, not only the parents adjusting and getting treatments to the kids, but also, for any rare disease that doesn't have a treatment, things are moving quickly. There could be a treatment at any time. And so the power of that, I know that is controversial, but just thinking about the power of being able to identify very rare diseases earlier, either in utero or just as an initial screening test for newborns, that could be incredibly powerful.
Wonderful, thank you. Okay, so we'll wrap it up. And again, Leslie, thank you so much. Dan, you want to just say something and parting words about the hub?
So thank you so much for coming, and we'll have more lectures, I promise. Hopefully we'll manage to even gather physically and have a bite to eat and maybe a beer and sit and talk science and progression and rare diseases and not so rare diseases.
Okay, thank you, everyone.
Thank you.
Have a great rest of the day. Glad it was
Thanks
quiet
for having
summer.
me.
Bye-bye.
Thank you.
Thank you.